In the 2000s, scientists recognized CRISPR-Cas9 as an adaptive immune system of bacteria and archaea, which these organisms use to fend off attacks from viruses. Emmanuelle Charpentier and her team described the RNA molecules that activate CRISPR-Cas9 in a study in 2011. A year later, Emmanuelle Charpentier and Jennifer Doudna published how exactly CRISPR-Cas9 targets DNA and how it can be used as a genetic tool to alter the genetic makeup. The discoveries that were the basis for the award are therefore just 10 years old. This unusually short period for a Nobel Prize is proof of the enormous importance of Emmanuelle Charpentier's and Jennifer Doudna's research. Researchers have subsequently refined CRISPR-Cas9 into a precise gene tool that can correct faulty DNA, much like word processing software. The technique, which is therefore also known as genome editing, can be used to investigate a wide range of scientific questions. Since it is also comparatively simple, it is hard to imagine a laboratory today not working with this method. CRISPR-Cas9 has not only revolutionized basic research, however, it has also become indispensable in medicine, biotechnology, and agriculture. For example, physicians around the world are working hard to translate CRISPR-Cas9 technology into therapies for diseases, for which there are as yet no treatment options available. Microorganisms with modified genetic material are expected to produce food and drugs even more efficiently. And agricultural crops whose genomes have been altered by CRISPR-Cas9 promise higher yields and greater resistance to disease and environmental stress.
Professor Emmanuelle Charpentier, welcome. Thank you so much for taking your time. Of course, I'm very excited to have a Nobel laureate here with me today. We're all excited. I guess we're just all a little bummed out that we cannot see you live on a stage, because this is not a usual Max-Planck-Festversammlung. It is a COVID-Festversammlung. But to keep things interesting, this is also not your average Zoom or Skype interview. Professor Charpentier is actually sitting in Berlin right now, while I am sitting in Frankfurt. And everything you see is top-notch green screen technology. So no efforts were spared to do this festive occasion justice. Now, due to the pandemic, everything everyone is talking about, it seems, is science, virology, epidemiology, which is unusual. But there are a couple of scientific topics that have always been hot topics in the public debate. And I would say genome editing is certainly one of these topics. So Professor Charpentier, together with Jennifer Doudna, you were awarded the Nobel Prize in Chemistry in 2020 for CRISPR-Cas9, a sort of genetic pair of scissors that allows you to make precise cuts and to edit the genome. So basically to rewrite the code of life. So to many people, this sounds incredible, but also a little scary. And we will have some non-scientists in the audience today, especially once this goes up on YouTube. There hopefully will be more non-scientists watching this. So Professor Charpentier, how would you describe your discovery to non-scientists?
Yes, so the discovery, as you said, it's about technology that edit genes and genomes in cells and organisms. But importantly, what is important that the audience understands is that it's initially about the understanding of a mechanism that exists in bacteria, and that allows bacteria to defend themselves against viruses. So I guess the audience is now familiar with pandemic, with SARS-CoV-2. Obviously, we can be infected by viruses. Any organism in the planet can be also infected by viruses. Bacteria can be infected by viruses. They have developed a number of defense systems. One of the defense systems is called CRISPR-Cas. There are multiple CRISPR-Cas systems. And in my lab, we were lucky enough to work on one of the systems that is called CRISPR-Cas9. And this is the understanding of the molecular mechanism behind CRISPR-Cas9 that led to this technology that works a little bit like a software. If you imagine a DNA as a text composed of sentences and words, and this CRISPR-Cas9 works as a software that can recognize a specific word, a specific sentence, and replace a series of words or a word by another series of words, or letter of the word by another letter. And this is what CRISPR-Cas9 can do on the DNA of any cells and organisms. So this is what you described, the possibility to edit the DNA and thereby edit the code of life.
So let's recap. You just mentioned you were actually studying bacteria. So I think before the pandemic, a lot of people didn't even know the difference between viruses and bacteria. So there's two different species, and bacteria can be also infected by viruses. So you were looking at those bacteria, and I think you were not specifically looking for a new genome editing tool. So you were studying how do these bacteria defend themselves against the virus? And then you discovered CRISPR-Cas. So it was rather unexpected, right? It was a surprise.
Yes and no, actually. I started my career a long time ago, nearly 30 years ago, working on genetic tools in bacteria. So for someone who's a bacteriologist, it's obvious to come to the point that working on certain mechanisms existing in bacteria will lead to novel gene technologies. All the gene technologies that have been developed over the past 40, 50 years actually originate from research done on bacteria and viruses. So a number of enzymes, of proteins, of technologies were really derived from bacteria and viruses. At the point where we worked on CRISPR-Cas9, we were interested in a class of regulatory molecules called regulatory RNA molecules. And we came to this defense system. From my side, it was quite obvious also based on what my colleagues had hypothesized, that CRISPR-Cas would be a source of discovering a number of proteins that can act on the DNA and the RNA. We were lucky enough in my laboratory to focus on a bacterium called Streptococcus Pyrogenes that causes diseases in humans. And this bacterium was coding for the CRISPR-Cas9 system. And this is a particular system which at the time was the most simple system existing in bacteria, focusing on CRISPR-Cas. And that was clear, at least for me, and that it could lead to a very neat genetic technology that could be used not only in bacteria, but specifically in human cells and other types of eukaryotic cells and organisms.
Yeah, it's so interesting to hear what you can learn from bacteria, and that you're actually, it's very common to discover these useful tools also for humans. So I think useful is an understatement, because a lot of experts were very excited when this came out, when this discovery came out, and people have been calling it a game changer. So what exactly makes CRISPR-Cas or CRISPR-Cas9 different from previous genome editing methods?
So compared to previous genome editing methods, CRISPR-Cas offers a simplicity of design, a specificity of the technology, and a possibility to really use the technology in cells and organisms where other technologies were difficult to apply. And this is, one has to say as well that in addition to the technology that is simpler to use, also, let's say, cheaper to use for the scientists, the technology needs other types of technologies, such as delivery technologies, to bring the CRISPR-Cas technology to the targeted cells and organisms in order to really perform the work of genome editing. So what is really important to mention is that the technology, this discovery has benefited from a large number of discoveries over the past 10 and 20 years with regard to delivery technologies, with regard to the possibility to culture grow a large number of human cells, primary cells, stem cells, also all the research behind the biology of organisms, also all the research that led to very thorough and very deep technologies to sequence the genome, so also imaging technologies. So CRISPR-Cas really has benefited from all these discoveries and makes it a technology that can be really applied in an unprecedented manner.
So for a lot of scientists or people who work in the lab, you just mentioned there are so many possibilities and so many areas that this method can change. So what would be areas of, let's say, everyday life that you, in your opinion, this new technique has most impact on?
I think for the everyday life, I would say that it is critical to mention the fundamental research. I mean, a lot of applications in biotechnology, in biomedicine, in agriculture originate from research done in the laboratory, really basic research, fundamental research. And this research is really about understanding the mechanisms of life in a deep manner. And this has always been going on with, thanks to the technologies that were developed prior to CRISPR-Cas9 and that would allow to modify genes and the expression of genes to be able to unravel their functions and understands all the complexity of life. So this is one of the first really main, I would say, direct application of CRISPR-Cas9 is understanding better mechanisms of life. Then you have a lot of applications now being developed in medicine, so indirectly or directly, also combining research and development with really the possibility now to develop a large number of screens that allow to find new targets for therapeutics and also develop models of diseases that allow to test therapeutics under development. The technology is used directly as a gene medicine. So from my side, I co-founded a company, a biotech company called CRISPR Therapeutics already back in 2013 that is developing CRISPR-Cas-based gene medicines specifically to cure certain blood disorders, combining gene therapy and cell therapy, and some patients are already cured and surviving after 6 to 10 months of treatment. So there are a lot of applications now under development in medicine and also for the production of safe crops. That is also an interesting application with regard to the challenge of climate change and the need to develop new crops that can resist the changes of climate.
Right, yeah. So genome editing in agriculture, this is one of the big topics for the next few years. That's also very controversial in the public debate, but it's a very important discussion to be had when we think about climate change and also sustainability. And So you obviously, things like agriculture, diseases that have previously been hard to heal, maybe there are new ways for therapeutics. And one very important aspect that you mentioned is fundamental research, which I just like to stress, because now during the pandemic, the public focus is on science, and we're very lucky to have vaccines that have been developed fairly quickly. And I think there might be this misconception that you just order a vaccine and then the scientists deliver. It's not like that, because it's also these mRNA vaccines. They're based on years and years of fundamental research. So that's very important. Before we have an important applications, we have to first understand. Now, you discovered CRISPR-Cas in bacteria, and then you initiated a collaboration with Jennifer Doudna in the US. Why did you choose this collaboration?
Yes, so actually I met Jennifer Doudna at a conference on actually small regulatory RNAs in bacteria. There was a session on CRISPR-Cas. I was giving a talk, so presenting the work of my team on the CRISPR-Cas9 mechanism. And she was also present giving a talk on describing the structure of CRISPR-Cas proteins from other CRISPR-Cas systems. And so Jennifer Doudna is a renowned structural biologist who did a number of very, very nice work on proteins that interact with RNA, not only CRISPR-Cas proteins. And at the time, I was very much interested in understanding better the structure of the CRISPR-Cas proteins, because we had an understanding that there was a large variety of these Cas9 proteins, and it was important to understand, let's say, the structure and being able to know what could be common among all those different Cas proteins, to be able to later on, after developing the technology, more allowing to provide to the users systems that are extremely small and that can be more amenable for gene editing. And so the collaboration started. And I have to say something very important, as much as, as Jennifer and I received the Nobel Prize in 2020. Behind you have young scientists, and we were lucky enough that the scientists of our teams, so specifically Martin Jinek and Christoph Schilinski from my side, were able to work in a joyful manner, a very productive manner, to perform the experiments that led to this discovery of the technology.
So maybe to all the scientists or researchers listening, how was the research process? Or was it rather smooth sailing, or were there some major throwbacks?
For me, it was relatively smooth, in the sense that I had my line of the development of the project with, a methodology which I believe a number of scientists have, whereby you know your long-term goal, and you go step by step with developing a number of hypotheses and plan A, plan B, plan C.
Yeah.
Of the fact that your plan A can change to multiple plans A2 and
Yeah.
B2. So this was the way we were starting in my own laboratory. I have to say that for me, what was challenging is that I was on my way from Vienna to Sweden, Umeå University, when I started with the project. Maybe it was good because it gave me a lot of inspiration. I was sitting long hours in Umeå in my office doing some brainstorming. So I have to say that, in contrary to a number of projects that were running in my laboratories, this one was quite straightforward, and all the experiments that were performed were really giving the expected results that I was expecting, and that also originating from a number of discussions with very few students. My team was very small, very young students, master student, first year PhD student. This I want to mention it because sometimes this is what happens. What helped me a lot is actually a long experience in working in different laboratories. I did 3 postdocs very fast, also working in bacteria, but also working in developing transgenic mouse models, working on developing genetic tools in bacteria. So I knew as well what was in need in the genetic field in eukaryotes. I knew what we could find in bacteria, so this helped me a lot developing the project. What helped me as well a lot is that it was also a project that was connecting what I was also studying when I was a PhD student, and also when I was specifically doing research in one lab as a postdoc working on phages, and in my early career as a PhD student working on genetic material, and so this has helped me a lot. It was a little bit for me a way to bring everything that I had learned and developed along my career and bringing this together. And knowing what was necessary in terms of collaboration, there was also collaboration with a group of Jörg Vogel, who was based in Berlin at the time, and what I could bring, what would be in need to develop and go ahead with the project very fast and in an efficient manner. So, yeah. Mm-hmm.
Yeah, I can't imagine how exciting it must be to be a first-year PhD student and being able to work on this project. It's amazing. So what would you say, how has obviously the Nobel Prize has gotten a lot of attention to you, your name, your lab, your work, but how has your work itself, maybe the everyday work in the lab, maybe changed since receiving the Nobel Prize?
Actually, I will say that it's more since 2013. My life changed tremendously. There are some aspects that I experienced with the Nobel Prize, which I experienced maybe even more in 2015, 2016, when I was receiving a number of prizes, and where I had to also do a lot of representations for Max Planck around the globe. So my life was a lot of traveling, a lot of receiving awards. So this was very transformative because I had little time for the research in my lab, specifically because there were different phases of my life at Max Planck since 2015 that took me a lot of time and that still take me a lot of time. I received, for sure, with the Nobel Prize, even more and more requests. So is flooded of requests.
More and more requests and always a little time for the actual work.
It's very difficult for my team because they have difficulties to let's say to understand how much I receive requests. It's really overwhelming. Having said this, I try to spend all the time that I can to be present in my lab, and surely with the pandemic, it also allowed me to stay a little bit more in my laboratory. I mean, my wish is for sure to be able to to be next to my team. This is the role that I have, obviously, at Max Planck. One should not forget that I have a role that is to lead a lab and to perform research. So I try to combine all activities. And it has been a non-stop journey, actually, for me since really the last 18 years, really nearly non-stop.
Well, so we're very excited to see you further succeed in the future with your work, and I'm very happy that we could steal you away from your lab once again, so you could give us insight to this important, impactful work that you do there. So thank you so much. Thank you so much for watching, and stay curious.
Thank you.
Thank you.